Hello! How is everyone? Are we okay? Ah, okay, we're good. This is like, well, for us anyway, this is the end of week 2 in that we won't see you until Monday.
Yes.
But amazing. And I hope that things are getting a little more familiar and comfortable as you navigate the course link sites and the IT troubles that are happening at the university.
About which we must not speak. The troubles that we will not name.
No, not allowed to talk about those. So, yeah, I mean, obviously, you've been very communicative at keeping us, and don't worry, we got you. Like, if something like that happens, we will make sure that you don't, pay the price of that, and that your learning is prioritized. And why is there a sloth? Okay. So, yeah. Anyway, all good, and we hope you're enjoying. This week's seminar is asynchronous, so it's the week 2 seminar. Week 3 is the one that you can do in the dairy bush, that you should really try to do in the dairy bush.
Come on out and join me in the dairy bush.
Let us know, right? Like, if you're currently in Iran, then obviously don't come to the dairy bush.
Oh, yeah, but don't do that.
Yeah. Okay.
Thanks for playing. You guys are really good at these things.
Yeah, thank you. That was fun. Yay.
And thank you. There's a couple of, we'll check that out, some music recommendations.
Oh, cool. Yeah. Canadian?
Yep.
And lots of diversity in artists.
This is Canadian and Indigenous.
There we go. I love
Tanya
Quebec.
Yep.
Yeah, wonderful. We will totally get her on there. Oh, and I think she was going to cover a Jan Arden song too, which is pretty cool. So Anyway, there was like a Twitter conversation where I was like totally personing out. Like it was good. Okay, how are you doing? We're stressed. You're probably stressed. Anything you want to add to this square to kind of share or not? Let us know how you're doing on all of this.
I'm just ready for this week to be over. I am, being away in the field with limited and rickety internet for 2 weeks. You know what happened when I left? Everything. It just kept happening and it didn't stop. It's very impolite. And I said yes to a whole bunch of things. And now, present me is very frustrated with past me.
Yeah, I hear you. I hear you. And
so frustrated.
Frustrated's a good word, right? Yeah.
Tired.
Yeah.
Squiggly green lines.
Tired. Need a nap.
Yes.
Yeah. So much work, right? Okay. So many of you are in university for the first time, because this is the fall semester, right? We're not the winter semester. Okay. So many of you are in university for the first time, and it is perhaps not what you were told to expect by most of you. Were probably just in high school, not all of you. So, you may be trying to navigate expectations with what's going on. And so there are some things that are going to upset you that I'm going to say. And one of the things that we notice, especially those transitioning from high school to university, is there is a very, let's call it a healthy grade drop, right? So most of us did really well in high school. We got pretty high grades, especially if you're in university now. There are all of these cutoffs and all of these things. And so when we take a look at the grades of the incoming group, there's like no variation, no phenotypic variation. You're all 85, right? Or 87, right? Like there's no variation there. And over time, in your first year, there's going to be a spreading out of that variation, okay? And it's totally normal. So what I will say is this, and some of you have been communicating with us. We love to hear from you. Don't stop doing that. What I will say is this. If you're getting grades that are 15 to 20% lower than what you're used to, just keep on doing the same things. You do not need to have a revolutionary change in the way that you study. Okay, that doesn't mean that you'll stay there. It just means that you're managing the stress of being in first year in your first 2 weeks, and things will get better for you. Okay, but please don't panic and don't allow those grades to like change the way that you do things or the way that you think about yourself. If you're getting grades that are much lower than that, then let's have a conversation about some studying tips or some time management or something like that, okay? But, you know, getting 80 on a quiz is freaking fantastic.
That's very good.
You're doing great. Do not change, okay?
There's some requests that we tell their parents. This rule.
Please tell my parents. Oh, yes. If you would like, we can write an email and share it with you that you can then share with your parents.
Hold on. We don't have to do that.
You can show them this recording.
Okay, let's check in at camera number 2.
Okay.
Hi, parents.
Hi, parents. We have to talk to you.
Yeah.
Your kid is amazing.
Yeah.
Your kid
Super
is trying so hard.
You should have seen the fill in the blank that they did earlier.
You need to give them a little bit of a break
Cut
so
them some
that
slack.
they can figure out how to do this in the right way that works for them.
Okay, back to camera 1.
Okay, guys, good. Let's move on. Yeah. No, eat breakfast. Oh my goodness.
God.
Eat breakfast.
I've heard it's the most important meal of the day.
I think Kellogg did that as a commercial.
Yes, and he said, "Don't masturbate." He was a bit all over the place.
Finish that sentence.
Okay, let's just say, go and Google the history of Kellogg's cornflakes and why they taste like limp scabs.
Okay, do not drop out. We'll make a bagel now. Okay, good. Okay.
Yeah, you're right. Lunch is the most one.
Yeah, excellent. Just eat consistently throughout the day in a healthy way. Thank you.
Graze.
Okay, good.
Yes, there's some people that know about which I'm speaking. Cornflakes.
Okay.
Hashtag cornflakes.
Cornflakes. Okay, good. Wonderful. So thank you so much for sharing. We really appreciate it. For those of you going, when are they going to get to the stuff and start teaching? This type of activity opens your mind and makes it available to learn some stuff. So bear with us. We know that some of you are impatient, and that's totally cool. We get that. Okay. So let's do some teaching.
Okay, so two last questions.
What?
Invest in meal drinks if you're lazy. Yes, for sure.
Yeah.
Or if you just love tasty smoothies. And the other is how are tests structured? You're going to see some examples you've already seen. You'll continue to see. The structures of the test, we'll keep talking about them throughout the term, always. But essentially, you're going to see examples of these quizzes, these polls that we run in class. Those are a lot like the quizzes.
Yeah. Yeah. Yeah, everything that we do is deliberate. They're like Easter eggs. Yeah. And we'll tell you that this is like an Easter egg, okay? We're also going to develop an asynchronous study tool that it seems to be quite popular. So don't worry, we got you. We got you. Okay. Cool. So we wanted to come back to this because we spent a lot of time on this, and when we collect valuable and meaningful data, it's important to respect the data and to make sure that we don't just forget about them, right?
And it's also important to look at your data and go, did we talk about all those things, or did we think? No, we got so onto one track about some of the exciting things. We're like, but we forgot this other important thing.
This other thing. Let's look at these graphs. And what I'd love for you to do is to stamp the ones that you think probably have a normal distribution.
Stamp it.
Stamp.
Stamp like your life depends on it.
Okay.
Coolio.
Okay, this is great. Thank you. Okay, so clearly we can see or appreciate the distribution of the data on the left-hand side, right? But why are we stamping the blue ones then?
So some questions.
What's going on with the blue ones and our ability to appreciate the distribution of the data? Can you write on the sides or in the chat or something like that? Why are some folks stamping the blue ones going, yeah, probably normal? What's going on with that distribution? Why can't I see?
There's a great word. Ring-wu, skewed.
Yeah, nice.
The greater than 90 groups values together.
Yeah.
Yes. Those bins. It's all about the bins.
Some of the blue graphs have no data. You're right. Yeah. Good. Okay. So here's the important thing. If we're going to say one important thing today, this is it. Okay. The way that you present data and the way that data are distributed can be very different. A normal distribution is about the data, not the way that you present it, not your graph. So by having these, thank you so much, whoever circled these greater than 90 millimeter bars, you're artificially skewing the data. When in fact, and if somebody wants to do this for me, if we break down the last bar from 90 to 99, and then from 100 to 109, we would probably get a normal distribution. So we can't say for sure that the data are not normally distributed. Are we good? So the way that we I'll repeat, the way that we present the data and the actual distribution of the data are independent things, or at least can be. And one of our responsibilities as scientists is to use the power of visualization to accurately represent your data. And in this case, the bin size, right, the different sizes here aren't really doing that blue data set justice. Is that good?
Yeah,
does that make sense?
And I was just going to see if I could get the slide to update and teach it what to do.
Yeah. Okay. Cool. So it's probably normal for men, but the way that we graphed it isn't showing the distribution. Okay. So we cannot say that these data are not normally distributed. We cannot say that they are definitely skewed. Okay. But they look it because of the way that we're visualizing it. I hope that makes sense. Okay. Cool. If you have any questions, pop them in the chat. Otherwise, we will move on.
Who the heck has 90 millimeter plus fingers?
Fair.
Minute ball.
Yeah, totally fair. Okay.
And potentially Muggsy Bogues too.
Yeah, good. Here's what we're going to do. We're going to do some stuff. We're going to show you some animations. We're going to cover basically what I think is the coolest part of evolution. And we will touch on a little bit of natural selection, but we're going to spend a lot more time on the other stuff, which is great.
Which is important because I think probably to date in your training as a biologist, the focus has been on thinking about natural selection as a mechanism. Mm-hmm. And it's an important one. Like it's a brilliant, totally. It's a gorgeous one. Yeah. And we'll revisit it. But there's other things in the mix, in that great big potpourri of life.
That's it. It's basically all
This is how I stir potpourri.
Okay, it's all of the other stuff that we don't spend a lot of time on. So, just a reminder, veliger larvae, Glucidia larvae stuff. We're going to do some of that first, and then we're going to get on to the other stuff, right? Okay. So we asked you to spend some time thinking about the advantages and disadvantages, and then we asked you to drill down into 2 advantages and 2 disadvantages for each of these life strategies, right? And we want to kind of take that up a little bit and make sure that everyone is on the right track, or at least a good track. It may not be our track, but some kind of track, right? To kind of keep you moving along. So what we've got is a whole whack of spreadsheets for you to work on. Again, these spreadsheets are simply there to make it so that all of you can have access. If you click on one and it's full, just click on another one. It doesn't matter what your last name is. So just go ahead and do that. We're going to start for surnames that if you get on the A to L pages, we're going to talk about advantages of the Glucidia and the veliger. For those that are M to Z, we'll ask you to start with the disadvantages, so that some of you are working first on advantages, others are working first on disadvantages, and so then we all kind of can swap and share information. So we'll pop those links in the chat.
Links have been popped.
The
popping
popped.
has happened.
Okay, good. And we'll ask you to go and start filling out those spreadsheets. It looks like this. Probably. Is that what it looks like? Okay.
Tell me if you don't see the links, but they should be there.
Okay. There they are. Cool. I have a link? How do I get a link?
Join the chat.
Oh.
Also, if you scroll, now you're presenting. It's in the slide notes of this slide.
Yeah.
This slide.
Okay. I'll go there.
This should, if you escape out of the presentation view, this should update like the last one.
Oh, yeah?
Yeah.
Oh, that's cool. Okay. Do that. How are folks doing? Nice.
Yeah, see?
Yeah, that's good.
So you just have to, you can't do it, unfortunately, in view. And it's hidden.
Mm-hmm.
The next one is a different sheet, I believe. And then, yeah, do it. Do it.
Good. Wonderful.
Yay, links. Yes, especially if we're talking about cats.
Okay. Super.
In fact, that's actually a good natural history bit of learning. Never click on a links.
Oh my God. I'm just going to get a little sign.
They've got
Hashtag
claws.
dad joke or prof joke. Is it prof joke? Is that, yeah? Okay.
Hashtag funny joke, I think is what you're trying to say.
No. Okay. You're great, all of you. Super good. Let's take a look. So for the Glucidia, so just a reminder.
Glucidia.
Oh.
I just met a larva named Glucidia.
And it's grabbed onto my filaments of my gills, and I can't breathe very well at all.
Okay. So moving on. Let's take a look. The Glucidia are the ones that hold on to the fish gills, right? Okay. More active in production. Millions are made, sure. I think millions of the villager are made too, though.
Yeah.
So that might not be like something that separates them out, but it's sure. Making a whole bunch of them, definitely a pro.
Compared to lynx, for sure.
Okay. Gain protection from their host, totally. Yes.
Yes.
Love that one, right? Because they have a low chance of getting predated. Really good. Guaranteed nutrients, very good.
Yes.
Better survival rate.
Meal.
Yes. Very small, temporarily reduction of juvenile death. Yes. Good. Risk of Glucidia being lost or eaten. Sure. Much more energy intensive to acquire a host. Super good. Spread slowly. Excellent. More rapid reproduction. Okay. Very easily predated. Yes. Can easily spread. Yes. Exposed in water. Yes. Don't rely on a parent. So very good. More effective in larval production since zebras' dispersion is so high. Cool. Don't rely on success of a host. Very good. Yeah, these are really good. Let's see. Are there any other ones that are different? More difficult to get food. That's very good, right? Because that counters the other one. They need to find a host. Yes. Yeah, they can move upstream. I love that. Yes. In a way that the villager larvae can't, right? They become plankton, basically, and they float, right, with the currents. So they can't really move. Free floating, yes. They can travel longer distances. Sure, that's an advantage of that strategy.
Yeah, it's the same trait, essentially, or the same
Yeah.
contrasting of the same trait. And in some cases, it would be
Yep.
an advantage.
Yep.
And in others,
Good.
you've lost your habitat.
Yes. Okay. Yeah, it's a slower process compared with the villager. It's a riskier process for sure. So I'm looking at the Glucidia here that talks about, these different steps that they have to move through in order to be successful. And every transition from one step to another has its own risks, right?
Did anybody talk about investments
Yeah, somebody did. They mentioned sort of more energy being
Costly,
required.
yeah.
It's costly,
Nice.
right? Yeah, so that's really good. Okay. Amazing. Well done. Super.
I just want to call out a bit of biology that you remember from doing the pre-lab things. But the fact is, most of the species that have these strategies have no functioning eyes.
I know. They can't see.
Isn't that amazing? So if you put yourself in a room and said, I'm going to make a lure that looks like a fish, and you did it in the dark with no hands, just by, say, forcing your elbow to do it, I'd fail.
Yeah, but evolution's kind of good like that,
Yes,
right?
and if you take 2700 and we'll learn about the mantle. And the mollusk mantle, if given enough time and a little bit of a push, I think could become anything.
Yeah.
Like
An airplane.
Anything. Yeah.
Okay, cool. Awesome. So let's do, please don't stamp at all. And actually, I think I'm going to just like turn it off for a second, if you don't mind. No stamping. Here is a question, and it's an applied question. So remember that we are testing application when we talk about when we talk about topic tests. I'm going to put a poll on your screen. Here we go. Launching the poll. Good. And it should take you a few minutes. So we'll give you a few minutes.
And I'm going to be quiet.
Yes.
The whole time. Starting now. Starting soon. I whisper. That means it's not. They can't hear. Oh, you can't. I can't. Whoa. Doing a quiz stuff. You have to be quiet.
I muted you.
Blockidia.
Shh.
Arcadia? There's a murder at the Arcadia.
Yeah.
Stop. You haven't seen that show? You haven't watched it yet?
No.
It's very good.
Yeah.
Marty Short, Steve Martin,
Cool.
Selena Gomez,
Okay.
Friends.
Yeah.
Some people are saying half the questions are being cut off, but they are on the screen. Yes?
Yes.
Yeah. Did we turn off?
Okay, we've got about 75% of you have already registered an answer, so we'll wait just a little bit longer, maybe 30 or 40 seconds.
There's a couple of people that said they can't see the poll. Is that a platform thing?
Probably. Are you on a Chromebook? I think Chromebooks are the ones that have the biggest problem. And be careful, if you do have a professor that's using lockdown browsers, I don't think they are compatible with Chromebooks, and I think you have to use a different computer. It's a mess. It's a total mess. It works for me on Chromebook. Okay, no worries. That's why we have it on the shared screen as well. So if you can't click in, don't worry about it.
Just write it down.
Commit, though. Go, okay, I'm going to choose this answer. And just hold on to it. That's all that matters. Okay.
And then asking some other questions about if I'm working together with a friend, are my marks. These are not marks that are being collected. This is kind of a shared experience to practice. We're modeling what the next kind of marking outcomes might look like, right?
And it tells us where you're at so that we can adjust and all of that stuff. So let's end it. And I'm going to share the results. Now, there is some disagreement, which is totally fine. Don't worry about it. We're practicing together, right? We're studying together. So what we want to do is highlight a few things because these questions or these types of questions are perhaps ones that you've not seen before. Yes. Here we go. So here's what we've got. Okay. So these types of questions are perhaps questions that you've not seen before. And what we want to do is we want to talk about them because you're going to see them in our course. With biology, and I mean with other disciplines as well, but maybe not some like chemistry, there can be many possible hypotheses or possible explanations for something. And it's very unlikely that there is only ever one thing that's going on, like on the planet, right? Like there are situations where something is this way, but then in a different environment, it is this way. And so there are very few like hard facts in biology, but there are many plausible hypotheses or many, testable statements. And that's what we like to focus on, right? So here we've got, we're looking for a true statement. And we do not do things like what is more true or less false. We don't do that. That's horrible. So the statement is true, and the other statements are definitely false. Okay, always. At least that's what we strive for.
But you'd be surprised how long authoring one of these questions takes.
Oh yeah, it took us like an hour and a half to write this question.
Taking banks of them through years, and then having literally thousands of people look at them every year. Sometimes we're like, we have to revisit them just as you revisit your own data. We're like, that might be more clear, less clear. Yeah.
Okay, so let's work through some of these. Basically, though, we're looking for a true statement. Many times we ask you for the false statement. So what we want you to do, and some students get mixed up. They're like, oh, I read A, it was true, so I selected that. But then we're looking for the false one. So there are 4 true statements and 1 false. So what we want you to do in these cases is to evaluate each one individually as a true or false statement. And you can do that with all of our questions. Right? So the first one, you can go, oh, that's false. Then the next one, you can go, that's false. And then you can go the next one, oh, that one's true. So obviously we're looking for the true statement, right? So just evaluate each one. Don't scan until you find one. Just evaluate each one. That's how you do our questions. So if we can take a look at these questions, what you can see is there really are 2 that are in opposition to each other more directly, right? So one of them has to be false and the other one has to be true, or they both are false, right? So let's take a look, because those are the ones that you've been focusing on more so than the others. So species, so it's B and E. So let's take a look at B and E. Species that are dependent on very specific hosts are less likely to go extinct because they have highly specialized. Oh, no, I'm sorry. It's C. C and E. Those are the 2. Sorry. Species with Glucidia larvae are more likely to speciate because their populations are more easily subdivided. And species with veliger larvae are more likely to speciate due to extensive gene flow among populations, right? So basically, which one is more likely to speciate? Are the concepts that we've brought in, have they been applied in the correct way? Okay. So we take a look at E, species with veliger larvae. Veliger larvae are the ones that are distributed throughout the water column, right? They don't have a host species. And then it says that they're more likely to speciate due to extensive gene flow. And what we're going to learn, if you haven't picked it up from the readings yet, is the idea of speciation to become a different species requires a barrier to gene flow. Gene flow has to stop. So speciation is very unlikely to happen if there's lots of gene flow. Okay.
And remember, there's some questions, some of you asking and kind of revisiting these definitions in the chat about what is speciate. So essentially, you're talking about the process by which populations, say simply two populations of the same species, through time and some kind of division, some kind of impediment, evolve to become different species. So the verb there, evolve, not develop.
Right.
Evolve to become different species.
Cool. Okay. And some of you are like, how come I didn't know that? And how come other people knew that? Don't worry. We're not testing. We're just talking. We're just using this question as a means of having a discussion, flagging things in your brain. Okay. Good.
Some great questions. Do you want
to, you do there, I'll do here. Can I do C? Or do you?
Oh, sorry. Yeah, keep going.
C. Species with Glucidia larvae are more likely to speciate because their populations are more easily subdivided. So here we go. We talked about speciation needing to be isolated or subdivided, right? And so this, to me, is getting at the truth of how speciation might happen. Now, why are they more easily subdivided? Because they are at the mercy of host fish and where they go. So the fish, once they get infected with the Glucidia, go upstream, they go over here, and then some of them go over there, and they get subdivided in a way that the villager larvae that are just kind of like flowing in the current may not. Okay? Cool. And then for B, lots of you said B, species that are dependent on a very specific host are less likely to go extinct because they have highly specialized host attractant strategies. And this actually is not true. Those that have these like highly developed strategies for a specific host are really in greater danger of going extinct because of their reliance upon that single host fish rather than a whole bunch of different host fish. I hope that makes sense. We can continue these discussions in the discussion board after class, anytime. But we do have to move on, and I hope that's okay. And Smith will type furiously.
Yeah, because I just want to go.
Okay. Okay.
In short, some of your questions about speciation, it is not simple. It's elegant and beautiful, and that's why biology is harder than chemistry and physics. Because physicists agree about what a meter is, and chemists agree what a mole is, and species are hard.
Yeah. Okay.
Well, they're actually fuzzy.
Okay, so let's take a break, like a mind break for a second. Here's a fun video.
Full screen it.
Full screen it?
Yeah.
How?
Right there. So I cover the bottom one when I'm staring.
Yeah, okay.
Boo, dude.
So fun. Okay. Should we be serious again? Okay. We hope you're enjoying this. We think it's fun. Okay.
Yeah, I'm amazed by those things.
Yeah. Okay, let's move on. How do we not full screen this anymore? There we go.
Have you guys seen that thing before? I'm sure you've seen it before.
Okay. Okay. Sit back and relax for a little while, okay? And actually, there we go. Cool. Okay. Let's talk a little bit about variation and evolution.
Oh, it was too laggy.
And, oh, sorry. Okay. I hope some of you enjoyed it. Okay.
The link is there when you get the PowerPoint later on.
Okay. Here are a whole bunch of facts. Okay. You're going to like this. Genetic variation. You want to do these?
Yeah.
Go.
So let's talk about facts. So when we talk about genetic variation, just to make sure we're all on the same page, we are talking about at the level, remember the hierarchy, the biological hierarchy, we're talking at the level of DNA, and we're talking about among individuals within the same population. Alleles, same gene, different versions of that gene. The genetic variation within populations, it can change in various ways. And yes? Yes, Dr. Jacobs.
I have a question.
You have a thought.
I have a thought.
Yeah.
My thought is about the alleles.
Yeah.
Different versions, and I don't know the answer to this, different versions of the same gene. So that means different genetic code, right? So different genotype. Mm-hmm. But does it have to result in a different phenotype?
It does not, because what you remember about the genetic code is the redundancy built into the triplet, the ACGT, that when you have substitutions in the third base pair position, often they're invisible.
So wait a second. So if there are differences, even that don't code for a different base pair, they are considered to be alleles.
Allele can scale. You can talk about that at different levels.
Okay, yes. Thanks. I learned something.
So, and this is one of the things we talked about a second ago in saying you've been introduced to natural selection. So here you'll see it, natural selection, second there, but we've included it within a number of mechanisms. Can change in various ways. These are mechanisms. This is under the hood of evolution. Mutation. Natural selection, genetic drift, and gene flow. Mutation is involved in all of those things. DNA is involved in all of those things. But we'll break apart those mechanisms. Look at what it agrees. The primary source of genetic variation is mutation. It can also result from recombination, but mutations are errors, right? So this is why, this is again how biology is so important, and like life is like biology, is that errors you only learn. What is it in Zootopia when Shakira taught us that nobody learns unless they're getting it wrong? This is nobody evolves unless they're getting it wrong. Try anything. So mutations are errors, and that's how you learn. That's how we evolve.
Really good.
They do not. This is important for wording. This is, again, we've talked before where words matter. The specificity and the meaning of words matter. Yep. So the phrase that it evolved to, that's like big warning bells go off in my fuzzy head, because it's like, it mutated. And happened. And then
stuff. There were consequences to the errors.
Then evolution said yes and.
Yeah. Or nuh-uh. Sit down, little grasshopper. Okay. So yeah, this is really important, and it scales, right? So variation starts with mutation, and then there are consequences, right? And maybe there are no consequences. But what?
Somebody said, isn't Shakira in jail? And then they were like, yeah, try anything, just not tax fraud. It's like,
oh. So funny. Yeah. Okay, here we go. So here are some consequences, and we are not interested in you memorizing this.
You're going to start writing it down. But
don't write it down.
You're going to get it in a second.
You're going to get it in a second. Yeah. It doesn't matter. What we want you to write down is the idea that mutations can happen at different scales. There can be a point mutation all the way to a complete doubling or tripling of the genome. Right?
To massive parts of the genome that just kind of disappear. They're gone.
Okay. And these things also, the consequences also scale. Okay. Who cares? We can just keep on trucking. Right. To, oh my goodness, you're dead. And anything in between. So this is what we want you to get from here, right? Is that there are many types of mutations and that they scale. Okay, so take a look at this. It's interesting. Maybe there's something there that, you can link to something, but those were the big sort of key takeaways from this rather scary slide. Okay. Natural selection. And this is pretty much the only slide that we're going to have about natural selection, because again, we feel that you kind of got it. But what we want to do is we want to give you some key terms that we can then transfer to all of the other stuff. So we're going to use the word random and non-random. Okay. And what we mean by random and non-random is in reference to the outcome of what happens, the consequence with respect to fitness, okay? And this is really important because mutations happen randomly. They are errors. They're like, oops, right? Totally random. The outcomes can be random and non-random as well. Okay, when they are non-random, it means that they are selected. Okay, so if you go to a fruit stand and you pick out the best looking fruit, okay, because that's what you're going to eat, that is non-random. You are selecting, right, for the best looking. Same thing going on here. If there is a non-random selection for that oopsie mistake of mutation, then we talk about natural selection. Who's selecting? Nature, right? Because nature's awesome. So this is what we mean by random and non-random. Natural selection, you're selecting the fruit at the fruit stand. That is non-random.
Your fruit stand selections at the grocery store are very rarely random. You've got to talk to your roommates if they are.
Okay, good. So that's what we mean by non-random. We want to talk about the random stuff, basically the shit happens stuff, okay? Yeah. Because that stuff is cool. Yeah. So genetic drift is maybe a term that you're only just becoming familiar with as a thing. These are random differences with respect to fitness, okay? With respect to the survival of the species, of the individual, okay? So we're talking about being random, where things just happen. And what we want to do is we want to kind of show you examples of what that might look like, stories, so that you can go, oh, okay, that's what they mean by random, okay? So in order to do that, I've got to switch over to PowerPoint because I haven't yet figured out how to do it in,
Thank you, Michael Scott.
In Smith Point. Okay, because I haven't yet figured out how to do it in Google Slides. If any of you are Google Slide geniuses, let me know. But let's go here, slideshow from current slide. Woo! Okay, random, right? Genetic drift. There are many terms underneath genetic drift that we want to introduce you to. We're going to do it quickly. You can replay this, and we're going to give you some homework that will hopefully help you anchor those terms within your own brain and learning, okay? So genetic drift is basically evolution. Remember, evolution is about change. So change that happens by chance, randomly, with respect to fitness. So imagine, and this is where Smith is going to get all cringed and like kind of like sour-faced, because imagine that we have six-legged spiders.
Let's call them insects.
No, they're six-legged spiders. And we're going to pick one trait that they differ on. And remember, things don't differ by just one trait, okay? But let's imagine, we're going to simplify, that there are some blue six-legged spiders and there are some red six-legged spiders. And half of the population is blue and half of them is red, okay? So genetic drift, an umbrella term that we're going to dive into in a second, is basically random survival by chance due to what we call sampling error. So imagine that these are the spiders that survive, whatever it is, that pass on their genes. And over time, if we go like to time 2, what we end up having is a different composition or a different population, right? Of 44% to 56%. That is evolution because there's been a change in the allelic frequencies, but not for any reason, just by random chance, okay? Good. So if we asked you, is evolution happening here? You say yes. Is natural selection happening here? No. This is genetic drift. So what can that look like? Here's an example of genetic drift called the bottleneck effect. And the bottleneck effect is basically started off by a dramatic reduction in the population size for random reasons with respect to fitness. So imagine we have a 50-50 population and something terrible happens.
Dum dum dum.
Okay, good. Tornado comes in and like smokes a whole bunch of spiders, and we end up with a different representation or proportion within the population, 42% to 58%. Now, that was random with respect to the fitness of the spiders because it didn't matter if you were red or blue. If you were in the pathway of the tornado, then that's it for you. Okay. Oh, I even got that rhyming.
That was just a poem.
It doesn't matter if you're red and blue. If you're in the pathway of the tornado, that's it for you. Good. Okay. Now, so random. Okay. And just because we can explain what caused it doesn't make it non-random, right? It was a random process with respect to fitness. Now, here's another example of genetic drift that we're going to call founder effects. And it's very different than the bottleneck effect because what it is imagine we have a population of 50-50 and we have then a group, a subpopulation that moves away to start its own population. And they move, again, who moves? Is it random with respect to fitness? They go to the island and they start breeding. And we end up with a population of 29% to 71%. And it's random, okay? Wait, are we good? I think we're okay, right? Okay, I want to finish off. Okay, I want to finish off with gene flow. You've totally messed the leg.
Oh, sorry. Yeah, look at that. Okay. Wow, I really did.
Yeah. Okay.
Oh, look. It's thinking. What are you thinking?
We have 1 minute left.
Yep.
So gene flow. This is something totally different. Students get the words gene flow and genetic drift mixed up quite a lot. So make sure that you anchor that in your minds. And if you need to, during a topic test, you want to make sure, just Google it. You'll be able to be like, oh yeah, that's that one. And this is this one. Okay. So it's the exchange of alleles across at least two populations. Okay. So think immigration and emigration. Gene flow is connected to speciation. We need a barrier in order to allow for something to evolve on its own to eventually become maybe a different species. If you have gene flow, essentially you're dealing with the same genetic population, right? Did I say that in a way that makes sense?
Panmixia.
Yeah. Okay. So gene flow looks like this, right? We have one side 2773, the other side 100 and 0, and they're just like swapping genes, right? So, you know, they're going back and forth, they're swimming, or they're blowing in the wind. And over time, the population then changes, and you have to leave your genetic material there. You don't just go there, you got to breed there as well, right? And it kind of starts to mix up.
And look at that.
That's it. I think we're done.
We're going to show that
Yeah.
question or no? That was hidden.
No, let's not.
Okay.
Okay.
So
That's it.
It's strange for us to say this on a Wednesday. We won't see you till next week, of course. So take care of each other.
Yes.
Be safe. Have a great weekend.